Hey, I'm Lauren. I'm really passionate about biotech and neuro-oncology, and right now I'm doing a deep dive on how I can leverage AI to improve patient outcomes. So in this, I'm going to be reviewing a literature review coming with the University of Toronto, and they're looking into computational pathology approaches for intra-tumoral heterogeneity, and specifically going into how this plays into high-grade gliomas like glioblastoma and some other cancerous tumors. So let's dive into it. Okay, so like we said earlier, we're focusing primarily on glioblastoma, just because it is a good case study for understanding intratumoral heterogeneity and like the computational approaches, as I mentioned here. The really cool thing about this is in a lot of cancers we focus, and I highlighted this down here. Oh, it went away. They focus on understanding the molecular analysis that are either with single region or bulk based tissue analysis, but that limits how much actual information and content you can gain from having that heterogenetic application and analysis in and of itself. So from that, we're like it says even here, when applied to the entire cancer, including reoccurrence and metastases, it's really, really stunting the growth of having precision medicine approaches towards this. So in order to do that, we have to understand what's going on in the individualized location and cellular components of the tumor itself, but then also to how is this changing longitudinally, how is this changing spatially, how is this changing with like it said in the very beginning of the abstract, in infancy and in different age demographics. And then when you try to do a resection or when you try to approach with different treatment, status quo options, we could say radiation or chemotherapy, how does the minimal residual disease look like within that, after kind of putting on this one's not really focusing more on that. It's focusing more on the cellular composition and understanding what landscape from our current approaches to understanding cell sequencing are we not getting in the whole picture of this, and then what is that lack of knowledge doing to our full understanding of how we're approaching this disease and approaching our subsequent therapeutics and our subsequent development of new treatment ideas. So it's very interesting, but I do like this in particular, especially for a literature review. I really enjoy how they focused on understanding that it's more of a bigger kind of issue, aside from just being, well, in this patient demographic there's not this. It's more of no. And what we're seeing a lot, at least in the recent papers that I've done for this series, is really understanding how complicated and how much of a challenge this intra and intertumoral heterogeneity of these high-grade gliomas. So very exciting. They say how typically for multi-region sampling, this is something that needs to be a necessary adaptation, but where and what location of that could be subjective, and is not really in a standardized practice at the moment. But they were saying how through using computational tools with artificial intelligence and deep learning, we could obviously hopefully add some standardization and some tangible implementations for workflows or ways to have this as a pipeline from the image to scan pipeline, one of the other labs did. And I think that's a really good actual example of this coming into play. What's even funnier is within that last example that I mentioned, they say this paper was published before or this wasn't a paper review, but it was before last year, before the other one kind of came up. But it's so funny because they're saying we would need to possibly approach this with supervised classifiers and unsupervised methods to personalize the analysis. So it's very exciting that all these different kind of labs and approaches in this area are all on the same page, which I find is really cool, because that means this is we're on the right track. Here is some actually empirical evidence towards why the individual location of some tumors can't be fully captured with the current status quo that we're seeing within getting these profiling methods at the moment. So it says right here, so specifically in glioblastomas, there's a lot of molecular differences between case to case, patient to patient. And while like every case should have some heterogenetic like similarities, the prognostic differences and like differences in like how people are going to respond to therapy are very subjective based on those earlier, really small variations. So here, this is talking about our IDH1 gene, which we mentioned before. It's within a 5% of the population of glioblastoma tumor cases, but that shows prolonged survival, so 31 months over the common IDH wild type mutant, which is a 15-month survival after initial disease onset, I believe. So even that, like even having the IDH wild type, like subfield, say like that's like a heading that you want to put into one of your like extraction features or something. Like even this like really, really small genetic like mutation difference is like something that could really, really significantly, like, see, this is almost, it's more than twice, right? Like that's insane. So it's very, very important that we kind of understand how we can better do this. So it makes sense as to why like this is impacting the possible efficiency of current treatments, because we aren't able to get that full like understanding. It says like here, like, you know, understand the microvascular proliferation or like necrosis and why they would be more aggressive based on like different treatment options. So here we have like a different overview of the different pattern recognitions for like the cancer stem cell models, and then what it also has been in for glioblastomas. And the different diagrams of these are either based on data from like just understanding cell signaling, and then also understanding the findings that we get from the like single cell transcriptomics, which is for this one that we're able to see these different cell plasticity variations. And then for C, they use this laser capture approach to enhance the previous, like prior transcriptomics variations, looking into it. And then through there, you can kind of get a more like bigger picture onto like feature variation. Here you can even see it doing that with the localization from our original data from here, and that originally stemmed from this, which in our very first research paper review video, I kind of showed a little graphic of this, and it was explaining how for some, for I think it was pediatric like low-grade gliomas, there is this BRAF mutation, right? And that is a protein that comes on, and it happens kind of during this cycle. But then 4D, when I was mentioning the understanding the longitudinal heterogenetic like differences, that data is from here, which is, well, I mean, it's more so like theoretical, but it's saying you could kind of understand this evolution longitudinally with like having these different tumor subclonal cell evolutions. And then within that, you can like figure out, so if these like treatment resistant cells are going to continue on past like intraoperative like resections and everything, past like first line, second line treatments, then obviously the like reoccurrences would be more likely. But then like how, like why does that happen? Like they said here is still not well established, but I feel like with the new approaches that they're explaining here and what we saw in some of the other papers, like using these deep learning approaches, using these computer vision approaches, understanding the full landscape also from like a genomic side and breaking it down, doing like whole slice image extraction, and then applying these different self-supervised and unsupervised learning approaches, I think we're getting way closer to actually having a much more like reliable final product of like understanding more of these tumors. As I say here, while it is giving some like indication for like clinically relevant like outcomes and like possible positive patient outcomes, just defining like research to one of the single cell like transcriptomics section is not good. Within this, having a single transcriptomal subtype is like kind of too simplistic, and like we looked at earlier, having this like really complicated intra and intercellular like heterogenetic variations, that's like we need to level up to the next kind of complexity of our cell sequencing variations and cell sequencing techniques. So this is where having this hierarchical and single cell level heterogenetic variation or heterogeneity, and then here what I was talking about even in our first diagram, which here shows it. So here is like intra, and then we have our longitudinal heterogeneity, and then that's kind of a picture within the reoccurrence. And then multi-region reveals the spatial, which is like within that, see how the variation between these multiple locations. And then on here, you can see the longitudinal variation that's a result of this, which is a result of that. And then, anyways, going back to this. So understanding that having subtypes and genetic events that are carried out from the same tumor, not regionally defined, so or confined, and that's what's happening without having like a spatial or longitudinal approach. And here it says these results were recently confirmed by a multi-biopsy sequence, which showed that even within single cell profiling methods, they were influenced by sampling the tumor periphery versus the core. And within that, you're still having that knowledge gap of understanding like the traditional like tissue profiling approaches. And then here it's like, even though we do have like advanced single cell profiling techniques, like it's still not enough to kind of get the like representation from the other regions. So here's where they're diving into the having more precise and anatomically driven techniques. And then laser capture is one of them with microdissection. And then they also used mass spectrometry, and then which was based proteomics, and uncovered that there is spatial separation within these regions, and then that is programmed within like the proliferation, migration, and hypoxic response. And then within these, you can understand what are the different supporting like variables that would like drive some of these microenvironmental factors. So here too, it's saying this same kind of like pattern can be extended into immune cell interactions, and it also can like play a significant role in the phenotype and the like prognostic of the special tumor or of a specific tumor. So here it's talking about promoting tumor growth by suppressing effector T cell functions. And then, so it's kind of like with CAR T cell therapy, and then stimulating tumor proliferation through secretion. And it's talking about the macrophages having a causal role in inducing the these proliferation and these pro-tumor growth suppressors, and understanding here that through inducing this, it still is localized to like that area of like spatial tumoral heterogenetic factors and everything. So when having like a generalization of just like tumors and like not understanding how they infiltrate like the surrounding cells is a very like undesirable problem to be having, because again, you're still not getting like a full picture. It's almost like you're getting a little snippet of like a little, say like you have your full vision. This is just like an analogy that I'm going to use, but you close your eyes and someone's like, oh, like squint and like turn your head, you can kind of see whatever image they're trying to look at. This is again an analogy. You can't totally see that image. I feel like that's more of like the approach that we're taking with these tumors. They're trying to squint and try to kind of see it a little bit, but we're not fully getting the full picture. And then when you open your eyes, you're like, wow, I can totally see that image so much clearer, or you move closer and you can see it so much clearer. And that's what we're trying to do with the computational approaches. Diagram is I really enjoy. It has the breakdown of like genomic, transcriptome, proteomic, and the phenotype, which is like with the central dogma of bio, and their rapid cell like turnover. And then within that, you can see the different like levels for analysis and histomorphological like phenotype variations and molecular changes, which is pretty cool. Possible way to combat this is to sample workflows from different single and multi-region sampling like case groups, and then you can see within here the differences within these variations and like how the subclonal and clonal cell populations are associated and differ with drug sensitivity. It could be like leading to chemo resistance to like chemotherapy and other drugs. So having like this collection of environmental interactions between these cellular states in the tumor is a really good picture into understanding the like regional guidance in radiological or microscopic like feature variations. So within this, you can see here, which is, this is actually the havoc. Like they basically kind of came up with one example where you could take this histomic analysis of variations of cancers. It's kind of like, we saw the cancer atlas of like all the genomes and everything like that. One of like the famous kind of online data sets of like when they sequenced all of it and have like a huge collective base. But now you can understand deep learning to extract the different features and like the vectors. So here's basically like a huge example where they show, this is all figure 4, let me try to zoom up, the different like histomorphological and molecular correlations in glioblastoma. So from A to B, we have our giant cell of the glioblastoma, and then H2, what does it say? Or here it shows our, sorry, I'm trying to read this into cellular astrocytes with numerous like bizarre multi-cell nucleated giant cells. And then for B, you can see like the diffuse nuclear mutated, staining pattern. And then within C, you have your spindle cell sarcomatosis component on the left, and then your histologic images of the gliosarcoma showing the biphasic pattern with the spindle cell carcinoma component on the left. The D is our reticulating stained cell. And then again, basically, as you can kind of see, but like it explains in the description, but typically this is like very hard to carry out on its own. And you can even see all these different samples that you had to get all the different regions of variations and understanding these patterns and these specific like transcriptional subtypes and subclonal regions are like so crucial. But through having this, you can actually use extraction feature for deep learning. So here using, they basically had this workflow from like high proliferation rates and then low proliferation rates. And then here they trained on a CNN, convolutional neural network. For those who are unfamiliar, it's a deep learning approach, and you can label patterns. And then they used supervised machine learning, again with CNNs, to detect the objective image and analyze that with the whole slice slide images, which I'm pretty sure, yeah, you can kind of see here something they're doing. And then here is having the example of them, and then you want to stain them. And then one of the data sets that they would use would be marked by professional or like an expert reviewer. So that could be like a pathologist who are like very trained in the actual like labeling and orientation of these different regions. And just to ensure that our training data is being input with like actual correct case examples, because when you're training machine learning models, if you don't train it with like some type of representative data that it could learn on itself, might not get the most accurate results, but that's not an issue in any of these cases. And if anything, putting the self-supervised learning approach actually solves that like pretty much there. And then here it says it exhibits based on having the benchmarking curve, which is the receiver operator characteristic curves, and that's the computational output that you're getting. So it's compared to the ground truth and then the performance success of the CNNs that are benchmarked, which again, convolutional neural networks using like the common metrics, which is this ROCC here. You get that based on the probability score, and then that is testing and the application, and these are different than the initial whole slice images that they're putting in, because again, it's trained on this data, gives you the analysis, and then you put it into this workflow, gives you this, and then gives you your basically like prognostic. And even this too would be easy to, I mean, maybe not easy, but it would be something that you could make into like possibly maybe patiently available. I'm not sure. I'm not in charge of that, so do not take this as like I'm just, this is all for learning, but that could be kind of something interesting in making it to understand and inclusive of all patients, and helping them like kind of understand more of what's going on. And it could even just help make more informed treatment decisions, and it could even help like when you're trying to see specialists in different locations, and maybe you live in an area that is quite far from a lot of like precision specifically cancer institutes or research centers, and then having this data with patient permission, of course, sent to your other specialists and your other healthcare providers could be a really good example of understanding how these cellular variations are like being impacted by like different treatment options and different like non-treatment options too. Again, this alludes to what they were talking about in the beginning of not having a good picture of understanding the whole like sequencing between these cells, and understanding what like sites could vary, and what techniques could like be positively or negatively influenced, I was gonna say impact, influenced by these applications. Here it's talking about how supervised computational pathological approaches are an aid, and obviously like it's important to like, while it could objectively like exceed the objective recognition capabilities of human observers, it's again like something that we should shown on equal rates they can outperform. But like deep learning pipelines are trained to learn patterns of interest by providing large data sets of human annotated images as ground truth, again what they said earlier. So through having that trained label, then you can associate the non-overlapping spatial patterns with the pixels. So that's like when you have like, say, a picture of a dog, a picture of a cat, you're like, you obviously have to have a picture of a dog and a picture of a cat. I mean, you don't obviously specifically, but I'm saying just to train these models. And then you put it like overlapping, it's like, what is this? And the machine is trying to learn. But if you don't actually have the like indicator of saying, okay, objectively, this is X and this is Y, and then telling the machine that, it's like your end result is obviously going to be undetermined or something like not indicative of if it's there, if it's not, because the training wasn't able to indicate the like feature without it, feature with, if that makes sense. But again, like I might be getting that incorrect. I'm speaking very objectively, and I'm all for learning. So if any of like this is being interpreted incorrectly, like please feel free to jump in the comments or shoot me a message. And I'd love to learn and improve. But yeah, from what I understand, the specific labeled annotations are super crucial for like associating non-overlapping and then overlapping spatial patterns with like labeled annotations. Here is saying like with workflows, it's adapted to learn molecular features that can accompany cancer cohorts, genetically defined ground truth labels, bulk tumor tissues, trained algorithms. Yeah. So here it's just, showing that there can overcome the patient-to-patient heterogenetic variations, but also too, it's like doesn't rely on morphogenomic dependencies, which may not always be shared across individual cancers. Important to note. They're talking about using unsupervised approaches, which again, this is having the deep learning. Okay, so I was talking about this earlier, but I was confused. So this is having the DLFV, which is basically having deep learning feature vectors. So having like signature image patches from like the whole sliced images, and then having that, you can use that. It's basically how the convolutional neural networks are going to classify the images into individual like activations of the features. So you could say like the histomic signature, similar to like what a genomic one and a molecular profile would look like. And then within these image patches, they can carry out unsupervised clustering and map the group phenotypic heterogeneity into regional precision using histology. And then this again, micro dissecting, which we saw in some of the other approaches, using AI-defined proportions to have the large tumor samples. Again, that also translates into predicting changes. So this is a good example of kind of a workflow that they used. And then similarly to what we saw in the deep learning pipeline, using like the transfer X learning and self-supervised learning approaches, this kind of alludes to a little bit of what they did there. Having the pre-trained convolutional neural network, and then having that serve as like the morphologic feature extractor and the deep learning feature vector. It's like very interesting to see how these can cluster and monitor the spatial coordinates of the tiles. And then it can also be used to visualize spatial heterogeneity. Here again is having the mapping, which is very cool too. This is super exciting for me though, because it's really like, I feel like very transferable to like clinical settings and like prognostics and patient outcomes, and also like therapeutic, like this whole thing I hopefully hope to see as a key kind of part in like creating like a treatment plan or something. And maybe one day if I ever become a physician, I can, have these work through these different workflows for different patient case variations, and we can move much faster and treat patients much more effectively and hopefully help beat this disease.